Hello, we are Thursday group number 3 for the Genetics Virtual Fly Lab. Some background information is that the mutation we were assigned was named Z, which had the phenotype of dark brown eye color. The mutation we studied was found in the model system of Drosophila melanogaster. This model system is used because it is a great resource of population genomics and quantitative traits. As part of the lab, we were given a mutation with 100% penetrant recessive phenotype. And our goal was to assign the mutation to a chromosome using a balancer chromosome with recessive markers. We could have assigned the mutation to the X chromosome or chromosome 2 or 3. Balancer chromosomes have many purposes, including to maintain deleterious mutations, prevent the recovery of recombinant gametes and crosses, which is what it was used for in this lab, and to assist in the mapping of unknown mutations to a specific chromosome. The procedure consisted of many steps, including mapping to a chromosome, which we used the balancer chromosomes to perform crosses for chromosomes 3 and 2. The next part of the lab dealt with mapping on a chromosome, where we performed a 3-point cross. And finally, we performed a complementation test. Major questions that were asked during this lab was 1, what chromosome is the mutation on? 2, where is it on the chromosome relative to the things I already know? And 3, is this mutation an allele in a known gene, or is it an allele of a new gene?
When trying to map our mutation to a chromosome, we can first remember some useful background information. This includes knowing that when two genes are on the same chromosome, F1-F1 crosses will result in the F2 generation having a 1:2:1 phenotypic ratio. When two genes are on different chromosomes, the F1-F1 crosses will result in the F2 generation having a 9:3:3:1 phenotypic ratio. This 9:3:3:1 phenotypic ratio results from independent assortment of the two genes. If our mutation is on the X chromosome and the mother has the mutation, then all F2 males will have the mutation as well. We were told that all of our simulated F1 results were wild type. In this table on the left, we crossed males which were known to have the B balancer with unknown females. The B balancer is found on chromosome 2, and here U is representing our unknown mutation. So in this U cross B crosses, we got a 9:3:3:1 ratio in F2, which implies that independent assortment took place and our mutation was not on chromosome 2. In the table on the right, we crossed males which were known to have the E balancer with unknown females, and we got a roughly 1:2:1 phenotypic ratio in our F2 generation, which implies that the two genes were linked on the same chromosome and our mutation is on chromosome 3 because the E balancer is the balancer for chromosome 3.
After determining what chromosome we believe our phi mutation was located on, it was time for the 3-point cross. Here is presented the possible crosses between the chromosomes included in the study, which are chromosome X, 2, and 3. Below is a table describing the phenotypes for each mutation. It is important to remember that plus designates wild type and a capitalized first letter indicates a dominant mutation. Table 1 is a summary of potential final crosses that would be completed in order to generate the F2 flies. Here, we wanted to cross the fly in F1 with a phi that is homozygous recessive for all genes. This way, if any of the phenotypes are the wild type phenotype, we will know that it came from the first phi in F1 and not the second phi in the table. Since we also know the locations of the wild type alleles on the first phi in the table, we can figure out how many crossovers occurred by the phenotype of the offspring as well. We would use females of the F1 genotype and cross them with males that are homozygous for all genes. we need the crossing over to occur within the F1 genotypes so that we can determine the location of the genes. Here you see the crosses between the parent, for the parent cross with homozygous male and female, which then is used for the second cross from the heterozygous female with the homozygous female, and below is the possible phenotypes for the F1 cross. Now here is our completed table of the 3-point cross, and when comparing the non-crossover and double crossover phenotypes, it is determined that the Lyra wing mutation is in the middle. Here above is a representation of our chromosome model, and below is a table determining the order of the mutations. In order to calculate the distance between each gene, the percentage from the table previously is used from the single crossover plus the double crossover in order to determine the distance between each mutation. So here you can see that our unknown mutation is 14 mu apart from the Lyra wings mutation, and the Lyra wings mutation is 17 mu apart from the stubble wings mutation.
From the information in Flybase, we were able to determine that the Lyra wings gene is 40 map units away from the end, and that the stubble bristles gene is 58 map units away from the end. When this is paired with our information about the double crossing over, then we know that our mutation codes for sepia eyes, especially when we know that our phenotype is dark brown eye color. So for the complementation test, we hypothesized that the gene for sepia eyes was mutated. When a cross between genes mutated within our unknown is occurred, then that would be sepia1 crossed with our unknown gene. That would result in sepia eyes. No complementation would occur because they are in the same complementation group. However, when we conduct a cross between our unknown and a gene we hypothesize is not mutated in our unknown, such as our unknown with scarlet eyes, then the mutations, since they're in different genes, would cancel out and result in wild type offspring.
Based off the data we collected in our lab, we can conclude that our mutation Z is on chromosome 3. We can conclude this because of the 1 to 2 to 1 ratio when crossing U and E.
As saw previously, you can see that our mutation is located 26 map units on chromosome 3, which is found before the Lyra wings gene and the stubble bristles gene. And this represents the sepia eye gene, which is also determined with our complementation test between the Z and the SE1.
So for further directions, the next logical step is to research this mutation and ask the question, how can the sepia eye color mutation be induced?
We would use PCR to isolate the area that codes for the sepia gene, and we would see how different types of mutagens affect it. We would have 4 groups. One would be a control group, one would be exposing it to X-rays, another would be exposed to an alkylating agent, and another would be inserted with a transposon. This study would help us understand the effects of specific mutagens on the gene, as well as what allows the control group to develop when there are no outside forces influencing it.